Well, good evening. Good evening and welcome to the 2017 Hagey Lecture. My name is Ian Orchard and I have the privilege of being the Vice President Academic and Provost. Let me acknowledge that we are on the Haldeman Tract, the traditional territories of the neutral Anishinaabe and Haudenosaunee peoples. Let me welcome faculty, staff, and students of the university, and also those of you who have come from the broader community. Welcome to the University of Waterloo. We're delighted to welcome so many people to this very important lecture series. These lectures are sponsored jointly by the Faculty Association and the University of Waterloo. And I'm delighted to have this partnership with the Faculty Association, who show such leadership in enabling the university to achieve its overall ambitions. The Hagey Lectures are the premier invitational lectures here at the University of Waterloo. This distinguished lecture series was established in 1970 to honor the contributions of Dr. J.G. Hagey, one of the founders of the university, and its first president in 1957. So we're now celebrating Waterloo's 60th anniversary. Also, of course, J.G. Hagey Hall of the Humanities is named in honor of him. As I mentioned, these lectures are sponsored jointly by the Faculty Association, who are also celebrating their 60th anniversary. A committee of faculty members is formed each year to administer the Hagey Lectures, and these lectures always bring outstanding individuals whose work has cut across traditional disciplines and whose contributions have profound implications on the intellectual discourse of our times. Individuals who have distinguished themselves internationally in scholarly and creative endeavors to exchange ideas with faculty, staff, and students, and to present a lecture that will challenge, stimulate, and enrich not only the University of Waterloo community, but also members of our broader community. Hagey lecturers have included David Suzuki, Margaret Atwood, Margaret Macmillan, and many more names that have scrolled across the screen this evening. And tonight, Carol Barnes. So let me thank the members of the committee for their outstanding work and their selection over the years of these outstanding people. Now it gives me great pleasure to welcome and introduce John Mielke from Applied Health Sciences and former Hagey Lecture Committee Chair to introduce our speaker, Carol Barnes. John.
All right, so good evening, and let me welcome Professor Orchard's earlier welcome to the 2017 Hagey Lecture. As you might imagine, introducing a speaker for a lecture series with a nearly 50-year history can pose something of a challenge, and I spent a fair amount of time trying to decide the best way to proceed. Initially, I had thought that I might take a very traditional academic approach and mention that this evening's speaker has contributed to the publication of more than 200 papers and several book chapters, many of which employed fairly sophisticated electrophysiological and behavioral techniques to help us better understand how a relatively small temporal lobe structure known as the hippocampus processes a vast array of sensory information in order to form memories. Upon reflection, though, I thought that such an approach might be regarded as lacking in creativity. After this, I thought that perhaps I would take the approach of noting how she had served as the president of the Society for Neuroscience, the world's largest organization dedicated to the brain sciences, that she was integral in establishing the Evelyn McKnight Brain Institute at the University of Arizona and continues to serve as its director, and that the depth and significance of her work has been recognized by a broad array of awards, including election to the Royal Norwegian Society of Sciences and Letters, election to the American Association for the Advancement of Sciences, and receipt of the Gerard Prize, which acknowledges a person who has made a considerable lifetime achievement within the neurosciences. However, I decided against such an approach for fear of sounding something like a publicist. Running out of ideas, I even thought that I would take the rather unusual route of mentioning how she gained first-hand experience with Canadian winters during her years as a graduate student at Carleton University in Ottawa, which I suspect might have played a small part in her decision to only accept academic positions in the southwestern United States. That she relaxes, believe it or not, by gliding around in unpowered aircraft, and that one of the early motivations behind her work was a request that her mom made to help better understand her ailing grandfather. For fear of being too personal, however, I discounted this approach as well. Finally, after all of my reflection, I decided that the best approach would be to simply mention that Professor Barnes has done two very special things over the course of her career. First, she looked out upon the same sort of things that everyone encounters when they examine mammalian aging, but she was able to see things that many others missed. That is, instead of simply accepting that memory loss and dementia are typical outcomes of aging, she endeavored to determine exactly how the brain changes during the aging process and what the consequences of these changes may be for the way in which we use information to create memories. Second, she has worked in a tireless fashion through her research efforts, through her mentorship of students, and through her advocacy work to ensure that we understand that efforts to address neurodegenerative diseases such as Alzheimer's disease will always be found wanting unless we first understand the features that define normal brain aging. On that note, I'd like to end my piece and ask that you join me in providing a very warm welcome to our 2017 Hagey Lecturer, Professor Carol Barnes.
Thank you for the invitation to speak. To join the group of lecturers that you've seen flashing across the screen is just really thrilling to me. It's a great honor to be here. Now, there are many things that I could present to you today about memory and the aging brain, and you've already heard about my long experience with thinking about aging brain and my long study of it. But I needed to focus and choose just a few things to share with you tonight. And by its very nature, this lecture series and audience is by design very, very interdisciplinary. So I invite you to broaden the discussion after my talk in directions that you may want to hear more about, and I'd be happy to elaborate on things that are more of interest to you and that you want me to say a bit more about. So I look forward actually to the discussions afterwards. For this talk, I'm going to focus or try to discuss what we think is happening to most of our brains as we age, and what this means for memory across individuals' lifespan. In other words, what is a healthy brain when you're 40 or 50 or 70 or even 90? What is the normal variability in a brain and cognitive function over the lifespan that should be expected? And when does this kind of change in cognition cross the line really and become a concern? Actually, as you've already heard, I first became interested in the aging brain and memory when I was in graduate school in Canada in the early 1970s, first at Carleton University in Ottawa, and then I moved for my dissertation experiments to Dalhousie in Halifax, where I finished these experiments. And this time span of 45 years has allowed me to see that we've really come a long way in separating myths and facts about the aging process. And I'm going to structure this talk around that experience And to illustrate this, I'm going to take you through what I believe to be transformative advances over the past half century in our understanding of the impact that aging has on cognition. So first question that you might ask is, well, why is the transition to old age considered to be 65 in humans? Well, one answer, at least in the United States, is that the age 65 was statistically determined in 1935 by actuaries as the age of typical retirement of individuals at that time when they were setting up the Social Security then. And just to point this out, in the 1930s and 40s, of course, the average life expectancy at birth was 62 years age, whereas today in the United States it's 79, 78 for males, it's 81 for females. If anybody's interested in sex differences in longevity, you can ask me at the end. There's lots of interesting answers and questions there. But today, what I want to tell you is that 65 has remained the conceptual transition point into old age, and in fact is used to define what has been called a young old age group in most studies today across the world in aging brain and cognition. For Canada, in fact, the average life expectancy at birth is higher than in the United States. It's over 81 years of age, 79 for males, 83 for females. And when Mackenzie King introduced legislation that became the Old Age Pension Act in 1927, it was for those older individuals, Canadians, that qualified over the age of 70 years, okay? And it wasn't until more recently, till 1965, that the pension eligibility age was lowered from 70 to 65 years with the introduction of the Canada Pension Plan. Now, personally, I would prefer the definition of old to be 70 rather than 65, since I'm older than 65, but I'm not over 70 yet. But we're still going to use age 65 years for the purpose of this talk, because 65 is used as the age boundary for old in the studies that I'm going to be talking about this evening. So, as I said, we've come a long way over 50 years in separating assumptions or myths about the aging brain from actual knowledge about how memory traces are laid down in the connections of cells between brains. I'm going to make you all experts on the synapse and how memory is formed in the brain. I'm really hoping that you enjoy this experience. I think you will. While there's a lot of research being done on brain diseases that occur with aging, the reason I choose to study the aged brain in the absence of disease is because I believe that to tackle the brain disease, to tackle and understand brain diseases meaningfully, you need to understand what is underneath those diseases. That is, what are the changes in the aged brain. In other words, you can't hope to fix it if you don't understand what it is you're trying to fix. Simple as that. So, we are beginning to understand the details of how the brain stores information, or what the biological basis of memory is in the brain, and I hope to impart that to you this evening. Now, these are the myths. This is then, or the things that, see. This is then, and this is now, what we thought 50 years ago, what we think now, and I'll be stepping through these points to explain to you that we have made progress over these decades. Okay, so then the first of these myths is that if you become old, you become senile. Okay, but nobody now believes that somewhere around 65 years of age, senility sets in. Now, the most common forms of dementia are, of course, the two most prominent are Alzheimer's disease, which has a specific pattern of brain pathology that's very gradual in onset. The other is vascular dementia, which is caused by one or more strokes and is typically much more abrupt in onset. But even 50 years ago, the prevailing view of old age was that if you lived long enough, you would eventually become demented or get a dementing illness. But today, we know that that becoming senescent or growing Okay. All right, somebody's going to have to help me. We're stuck on C now. Okay. This is not the perspective you should be getting. So we know today that becoming senescent, which means growing old, is not the same as becoming senile. So not all of us are going to have Alzheimer's disease or vascular dementia. So we know that AD, Alzheimer's disease, is not inevitable. In fact, in a study that looked at a representative sample across, drawn from people across the United States, of those people 70 years or over 70 years, 14% have dementia. So it's only 14%. Now, only 14% is a huge number of people afflicted by dementia, but this means that 86% of us 71 or over are not demented. And these are the numbers from the study that I just cited. I think these numbers are pretty much equivalent in Canada and in most of Europe. These are the numbers from the study. Its prevalence of dementia for those over 70 years of age differs, of course, depending on the age group you look at. Overall, everybody over 70 is 14%, but 71 to 79 years, the prevalence is as low as 5%. 90 years and older, the prevalence goes up to 37%, and these break it down into different categories of the type of dementia. But now, most of us believe that even if one is reasonably free from disease, there are lifespan changes in memory. So that while 86% of us are not demented, we will have some change in our memory with age. Now, we already heard about the hippocampus tonight. One such temporal lobe structure that is involved, very important in memory, is called the hippocampus. And one reason for the focus on the hippocampus is that it's thought to be the main gateway to lasting memories in the brain. And we know that changes in this brain structure are an important contributor to age-related memory impairment. This picture is of what we call a rainbow transgenic mouse, where different colored fluorescent proteins label different subsets of cells within the hippocampus. And I think you can see this example as where neuroscience and art actually intersect. Okay. We know that the hippocampus is critical to lasting memories because if the hippocampus is damaged in humans or in other animals, memory is severely impaired. And one particular form of memory that the hippocampus is involved in is spatial memory, the kind of memory that allows us to find our way through the environment, to know where my Delta Hotel is from here, and I'm probably pointing the wrong direction, so you can see the function of my hippocampus. But we know the hippocampus is important, partly through studies. An example I'm giving you here is a positron emission tomography study in which the brain activity can be monitored by looking at glucose metabolism in the brain. And so this allows one to measure brain activity while people are in the scanner. So Eleanor Maguire and her colleagues in London had taxi drivers recall a route that they would take if they were told to pick somebody up, say, in Grosvenor Square, and then to drop them off somewhere else, say, Elephant and Castle. And when taxi drivers recalled these routes that they were thinking about in the scanner, the right hippocampus was powerfully activated. And this shows an average activation of the right hippocampus in an average of 11 taxi drivers at different levels of the brain. And this little yellow spot shows you the hippocampus. And so these are the kind of data that support the idea that the hippocampus is fully engaged in this type of spatial navigation. So what do we know about this kind of memory in aging? Well, in fact, it's been experimentally shown that the kind of spatial memory that depends on good hippocampal function does decline in aging. And this has been shown across species. That is, it's been shown to decline in humans, in monkeys, in dogs, and rats, and mice where it's been studied. So a summary then of concerning the thinking about the first point in my myth list concerning how aging results in senility is that while dementing disease is not inevitable, I'm afraid some memory change is. The example I gave you was spatial memory, but there are a number of kinds of memory that do change with age. And this happens in all mammals that have been studied. So because we know that spatial memory depends on the hippocampus, this provides an opportunity then to design experiments to discover what brain changes lead to these memory changes during normal aging. And so the second myth that I want to talk about, aging, you have extensive cell loss, so your brain is just, your cells are going away during aging. But nobody now believes that major brain cell loss occurs during the normative aging process as it does in dementing diseases. It does, you lose wide numbers of, lots of cells in dementing illnesses, but not in normative aging. So it's important to be reminded that the major cell type in your brain, they're called neurons, are born largely before your birth and die with you at the end of your life. They don't divide or re-enter the cell cycle like your skin cells or your liver and so forth. So this is a summary that of brain aging that was what was thought to happen to neurons during the aging process when I became interested in memory and aging in Ottawa in 1971. So that is the dismal picture where on the left are examples of neurons from young brains. They look great, big dendritic trees and so forth, but by the time you get to be 90, they're shriveled and horrible. But we now know this is wrong, which is a good thing. We have something to work with here. So there's really no major, oops, geez, I keep doing this. There's no major cell loss in the absence of dementing illness in the aged brain. So I'm going to give you some example cells, such as these three main cell types that have been counted across age. These happen to be rat cells, granule cells, pyramidal cells, but we have the same cell types in non-human primates and humans as well. And we really keep about the same numbers of these cell types in this important memory structure as we age across our entire lives. And more good news for those of us who are absolutely determined to age successfully is that the old cells are actually quite healthy. So in animals, it's possible to examine live cells, and in my lab has found that the properties of old hippocampal cells are similar in most respects to young cells. Old cells are quite healthy indeed. So you look at me and you say, okay, so normal brain aging, normative brain aging, if at least in the hippocampus there's very little cell loss, and those cells that are, that are there are quite healthy. Why do we have memory changes? What is it? Okay, well, we do know that at least two very important things, changes do occur in the brain, and that is reduced numbers of connections between cells and reduced plasticity. And what I'll do is start with the change in numbers of connections between cells, brain cells. We know that the brain has 86 billion neurons and it has 100 trillion connections between these neurons, and that axons of these neurons transmit information from one cell to the next, and most cell communication occurs in the brain by one cell sending electrical impulses down these axons, and then at the end of these axons are synapses, which are caused to release a chemical transmitter and across a small gap. which causes an electrical disturbance then in the next cell, and that's how information is passed on. And so while you don't lose hippocampal cells during aging, older humans and other animals do lose these synapses, okay, connections. Right. So plasticity, let's go on to plasticity. What is synaptic plasticity? Well, it involves the ability of one cell then to communicate with another through these synaptic connections, the synapses. And when a synapse becomes stronger, it produces, as I said, larger electrical responses in the cell that it's connected to. And it's believed that memories are stored as increases in synaptic strength in the connections made between cells. And although I've been able to show in my lab that synaptic strengthening definitely continues to occur in the aging brain, the strengthening is not as durable at an older synapse. I've been able to show that how durable this synaptic modification is correlates with how good your memory is actually at any age. So on point number 2, neuron loss is not widespread in aging, but there is reduced connectivity and plasticity that occurs in the aged brain. Okay, let me go on to the third myth, that brain aging equals passive deterioration. The brain just sits back and decays. Well, no one now believes that the brain does just sit back and deteriorate. There's been, I think, what I might call a fundamental shift in thinking about brain aging over the past several decades. We now think or believe that the brain does not passively deteriorate. Rather, in successful aging, the brain is constantly adapting in ways that suggest, in fact, outright compensation. And I'm going to give you 2 examples of this kind of compensation, okay? One animal experiment, one human experiment, all right. So for the animal experiment is one that I conducted starting with the following observation and question. We know that some hippocampal neurons from older rats have lost up to 1/3 of their synapses coming into the cell. The neuron requires a certain level of incoming activity before it can become altered and pass its information on to the next cell, like in the slide that I just showed you a moment ago. And in the face of this reduced input on the old cell, how do these cells manage to become active? Well, we were able to show that the way the hippocampus solves this reduced input problem is to make the individual synapses that remain on these cells stronger, more powerful. Okay? So fewer synapses then can discharge this cell. And this is evidence then for biological compensation during aging at a single synapse level. Now, at the network level, the observation that we're going to try to explain is that retrieval of memories in older persons of salient past experiences, say from 1 year ago, 10 years ago, or 20 years ago, often can be quite good in older persons. And we know that the hippocampus is involved in this kind of what's called autobiographical memory, remembering events from your past. And I'm going to describe an experiment that suggests that older adults actually can recruit additional circuits to achieve accurate recall as good as that of younger adults. So the experiment I'm going to tell you about is from McGuire and Frith, who did a study to look at brain activity during retrieval of these old kinds of memories while people were scanned using MRI methods. And when the participants in this experiment were asked to retrieve these memories of their life, past life in the scanner, you see that the left hippocampus is activated, becomes active in younger adults, while in older adults, to achieve the same level of very good retrieval, they used both the left and the right hippocampus to retrieve these experiences. So the neural circuits recruited for equivalent performance in young and aged individuals can be different. And this may potentially reflect adaptive compensation of the aged brain. It's not sitting back and passively deteriorating. Okay. So in summary then of point 3, the brain can adapt effectively during aging, suggesting actually great resilience and modification ability in older brains. All right, a fourth point. No one now believes that there are only a handful of genetic factors that probably can lead to either protection or acceleration of aging-related changes in cognition. There are many. We're discovering more every day. And I'm going to give you just a few examples. For example, having a particular variant of the gene KIBRA, you have alleles. You got one for your mother, one for your father. Okay, so you got different kinds of combinations of alleles in these genes, but having a particular combination in your KIBRA gene will make you have a good memory your entire life. You'll have a good memory forever. But for example, having another allelic combination of this cholesterol transporter, apolipoprotein E, if you have the fourth allele, this means that you're going to have a higher risk of contracting Alzheimer's disease throughout your life. And even at younger ages, people who carry this gene tend to have poor memories. Another example of gene that modifies cognition, the HDL cholesterol gene, high-density cholesterol. Genetic variants that produce high HDL levels predispose people to not only have strong cognition, but longer lives. I've been able to visit centenarian populations and so forth. It's really remarkable. Centenarians are people who live 100 years and over, but centenarians, their siblings, their offsprings, all tend to have high HDL levels. So a summary then of the fourth point, and while I've only talked about a few, this could be a 4-hour lecture, and I'm sure it's late enough tonight. A few points, but many complex genetic factors combine to contribute to your cognitive aging trajectory. Now, a fifth point, it was known that smoking, you might get cancer and so forth. There are a few lifestyle factors and risks that were known, but no one now believes that there's only one or a few. There are many, many factors that can either protect or accelerate aging-related changes in cognition. And among others, these include factors such as exercise, metabolic syndrome, inflammation, diet, stress, and social interaction. And I'll go through examples of each of these. For example, for exercise. We all know, of course, that exercise is good for maintaining good bone and muscle mass, avoiding osteopenia, sarcopenia, muscle wasting, or bone loss. But what's the evidence that exercise is good for brain aging? Okay, I'll give you one example from Gene Alexander's laboratory at the University of Arizona. Gene and his colleagues analyzed whether there's a relationship between the level of physical fitness and markers of a healthy brain. And they used magnetic resonance imaging to study 123 people of ages 50 to 89 years, and then they determined each individual's aerobic fitness score from treadmill tests, right? And they found that the better your fitness level, the more fit your brain appears to be. And as shown by this relationship between fitness and brain aging. So this is the fitness index, bad down here, good down here. So the more of the aging brain network pattern you have, it relates to lower levels of fitness. So if you weren't already convinced that you need a good reason to walk every day, this is your good reason to walk every day so that your brain is healthier. Now, as well as exercise, we also know a good diet helps control your body mass index and clearly has important critical effects on the brain. These data are from my colleague Lee Ryan's lab at the University of Arizona and indicate that as your body mass index increases, there's more brain volume loss, And you can see that here indicated in blue. And lower memory scores, so higher BMI, more brain volume loss, and actually these effects are really accentuated if you also have hypertension. And the effects correlate with memory as well. So you want to keep your weight as good as you can if possible, and you really want to control hypertension. So I'm going to talk about inflammation now. It's also clear that as we age, we really need to avoid, if possible, brain inflammation. So when you're skiing, wear your helmets. When you're cycling, wear your helmets. Actually, still, head injury is one of the best predictors of who is going to get Alzheimer's disease and who is not, especially if the injury is after 50 years of age. An interesting study, again, conducted in Lee Ryan's laboratory at the University of Arizona suggests control of brain inflammation is really important for optimal brain aging. Lee and her colleagues looked at two groups of age and education matched females, all living independently between 52 and 92 years of age. The two groups she split not by education factors or other things, but she split based on whether the participant had or had not used non-steroidal anti-inflammatories. NSAID users versus control. So they didn't use non-steroidal anti-inflammatories. The NSAID users had a mean group number of years of taking NSAIDs of 10 years, a minimum of 4. And within this age range, the control group, the red indicates brain volume loss. You see the red is areas where brain volume was significantly declined over this age range. And these areas we know are very important for memory. Over a similar age range, the NSAID user group showed dramatically less brain volume loss or change as measured by MRI, suggesting that there may be a protective effect of anti-inflammatory drugs on age-related decreases in brain volume. You don't even need statistics to see the differences here, much less brain volume loss. Okay. It's also becoming clear that the components of foods that are found in Mediterranean and other kinds of diets are clearly good for brain health. They have powerful anti-inflammatory properties. And as it turns out, lucky for us, foods such as leafy vegetables, nuts, dark chocolate, and drinks such as tea, coffee, and thank goodness wine, all have anti-inflammatory properties. And this is because they belong to a class of antioxidants called polyphenols. These polyphenols are contained in these plants. So it's particularly the flavonoids that have these anti-inflammatory properties, and it's the flavonols that are the predominant class of flavonoids found in, for example, dark chocolate or nuts. So dark chocolate, why am I saying dark chocolate? So if you want to know, milk enzymes actually degrade the action of the flavonols, and dark chocolate has more flavonols than the lighter chocolate. So anyway, that's why dark chocolate. Okay. In fact, newer studies have found correlations between certain kinds of memory and ingesting certain amounts of nuts, dark chocolate, olive oil, and actually coffee. So the good news is that just by including these things in your diet, you're undoubtedly helping to reduce brain inflammation. And through this, you're probably improving your synapses health, your synapse health overall. Okay. What about stress and the effects of stress on health and behavior? Well, these factors are becoming better and better understood. For example, it's possible to measure cortisol levels. This is the body's stress hormone in people who undergo MRI imaging. And when volumes of different cortical regions are measured, those people with the highest stress hormone levels also show reduction in volume, particularly focused on the frontal cortex thickness here. Now, this brain region is known to be very important for what's called working memory or executive functions. And these types of memory, such as working memory, show the earliest age-related changes. So for those younger folks in the audience, not good news for you, because working memory actually starts to decline in your 30s and continues down. And this one kind of memory change in normal aging is particularly annoying, at least to me. Okay, so an example is you're upstairs. You remember you need to get something downstairs. But the time you get there, you have no idea why you're downstairs or what it is you're looking for, because you've had multiple intervening thoughts, and it goes. So this kind of reduction in the frontal lobes caused by stress can worsen these kinds of memory problems. Tai Chi exercises have also been appreciated for some time for their stress-reducing properties. I just love this study, so I'm just going to show you the study. But it also is really good for stress reduction. And one concrete benefit is shown by the study, where one group of older adults completed a 16-week course of Tai Chi training, the other group a 16-week course of health education training, before being given a vaccine at this arrow here against shingles. And what the data showed was that the Tai Chi group showed an increase in cell-mediated immunity even during the training. So they got a boost in immunity even during training, and they had boosted immunity after the vaccination, which you see here, compared to the health education group. So this suggests that Tai Chi may have beneficial effects both for the brain and for the immune system. And of course, the brain and immune system are working together all the time, which we're studying more and more these days. Okay. Also, a lot more is being learned about the importance of social cooperation and altruism in the successful aging of individuals. And as we learn more about what structures in the brain are active during naturally rewarding situations using MRI, it appears that, in fact, behaviors that reflect social cooperation and altruism activate or exercise these brain regions, these areas of the brain, deep in the brain, that are responsible for reward. So an interesting study that was done showed that older adults who were studied over an 11-year period, I think, who, if these adults included in their activities volunteerism and altruistic behaviors, they had several associated benefits compared to those individuals who did not engage in these types of behaviors. And these are really quite remarkable. That is, they had better physical health, greater longevity, and increased life satisfaction. So to summarize in my fifth point, there are multiple life factors that can and do contribute to successful aging, just a few of which I've told you about here. So to summarize all we know, what we knew now and then about memory and the aging brain, Alzheimer's disease and dementing illnesses are not inevitable, okay? But some memory change does happen. There's minimal but selective cell loss. So because your neurons are with you from birth to death, you lose them, that's serious. But we're not losing a lot of neurons. That's good news as well. And the brain isn't passively sitting back and deteriorating. It's actively and continuously adapting. And we want to facilitate this adaptation process more and more. And there are many complex genetic factors that interact with lifestyle factors that go together to determine your cognitive, the success of your cognitive aging trajectory. So in conclusion, I think what I've shown you today is that the brain is remarkably adaptable throughout life. It's not just when you're young, it's throughout life. And there are many things that we can all do to assist in maintaining the health of our brains. And if there's one message that you take away from this talk, it should be that you should be keeping those synapses of yours active in order to close the gap between, if this is your lifespan and this is your cognitive health span, we want to close this gap between cognitive health span and lifespan. Exactly the kind of thing that you're doing tonight by coming and listening to me talk about synapses is exactly the right thing to do for your brain. Dr. Hagey was prescient, I think, in understanding where we would be today in our understanding of the aging brain is getting the community and everybody to participate in these kinds of lectures. So clearly, while we've learned a lot in the past 4 decades about the impact of age and cognition and the circuits responsible for these behaviors, there's clearly much more to be discovered. And the hope of all of us in this field is that we will be able to figure out ways to optimize cognitive health throughout the lifespan, and we're doing that now. One way, I think, to conceptualize the importance of this problem is that perhaps the only non-renewable resource in the world that's currently increasing is the number of people over 65 years of age. And so the grand challenge then is to figure out ways to keep us all functioning at the highest levels for as long as possible in our individual lives. Thanks for listening. I'm going to answer questions, so we'll figure out how to get this set up. Great. Do we have any questions from the audience?
I think you need to answer by the mic, just to make sure that it's picked up by the
I see. Yes. Yes. What's really interesting is there any dose relationship? So somebody was going to repeat these. Is there any dose relationship between the amount of NSAIDs that you take or anti-inflammatories that you take and how much protection you get? So there have not been any good studies that have rigorously, systematically looked, but even relatively low levels in some people were effective. So low levels for a long period of time. Just what's intriguing is that no NSAID use. Those are the people that showed the greatest changes. I have to tell you too, just to disclose all these, how did Lee get all these people that took NSAIDs for so long? We have this wonderful arthritis clinic at the UA. And so these individuals actually had arthritis and were using high levels. I know some people's stomachs can't take it, but even baby aspirin was effective, even though it's not an NSAID, yes. And we know our cardiologist friends tell us we should all be taking at least some aspirin for our hearts, to keep our hearts healthy. The drug Aricept, yes. So there's you have at the synapse an electrical signal, and that electrical signal releases a neurotransmitter that goes across the synaptic cleft and goes to the other side of the cell, the next cell, and activates it. So this chemical transmitter, it has an enzyme that breaks it down, okay, at the cleft. And so what Aricept does is let that transmitter hang around and activate that cell more. And so it's an enzymatic blocker. so that the actual chemical signal has more of a chance of getting to the next cell to communicate. Uh-huh.
Can you comment on the role of sleep in the aging of brain?
Oh, okay. I'm supposed to ask you to wait for the mic, but it's about sleep. Okay. Okay. The role of sleep in aging. Actually, I'm sorry. I'm breaking rules. Sorry. The answer is complicated because if you get too much sleep, that's really bad. If you get not enough sleep, that's really bad. So there's a study that came out that I was, astonished by, actually. Those people, it's a study that looked at sleep patterns and how much people slept per night, and then it followed people for years and looked at mortality. And people who were sleeping like 9 hours a night were just as likely to have high mortality as those that slept 5 or 6 hours, so 9 or 10 versus 5 or 6. So, I mean, the sweet spot seems to be around 7 or 8, but actually there are great individual differences. Those lucky people who only need 4 hours of sleep, and they've only ever needed 4 hours of sleep. There are some of those around, but they're pretty rare. So, also, sleep quality is another issue. Frequent waking and so forth, of course, and disturbance in sleep. But nobody has studied in a huge population of people to follow their sleep quality, so we don't really know the effect on mortality. But we do know that the better sleep quality it tends to correlate with better cognition. There's people up there. Can you help me figure out who is going to answer questions? There's people up there.
My question, I'm wondering in your studies with other mammals, if any of them have had the problems I've had with what they call baby brain loss or memory loss or menopause loss or now cancer brain loss. And how does that fit into your theory, and is there anything we can do about it?
It's a really critical question. I have a colleague, a very good colleague who's an endocrinologist who actively is doing an estrogen trial in Alzheimer's patients for keeping up certain amounts of estrogen levels. And there are studies out there, MRI studies and memory studies around menopause that shows that cognition can be affected by dropping levels of estrogen. So it's an important thing to treat, but of course we still don't have those compounds that only go to the brain, estrogen receptors, rather than going to other body tissues, which can be dangerous for cancers and so forth. So, but she is actively working on this area now, I know, and a lot of animal models are supporting this, and she's got some of these compounds in clinical trials right now. So I'm hoping she's going to have a good outcome, but clinical trials are hard. So.
Thanks. I was just wondering, I noticed in the pictures, some of the MRIs, the hippocampus on the left lit up, and in others, the ones on the right lit up. Is there kind of a category for the right hippocampus does this and the left hippocampus does something else?
Yeah. So for spatial types of memory, it's the right, and for autobiographical retrieval, it tends to be the left in young people. But as I showed you, older adults use both hippocampi to show accurate retrieval. So yes, the brain is lateralized. It's not only in humans, but also in non-human primates. It's lateralized as well. So there are separate functions of the two halves of the hippocampi, even in humans.
So I'm just wondering whether when those synapses become non-functional, is there anything that can make them become functional again?
Yeah, and so these are the kinds of therapeutic questions that are hot topics for drug companies, because if we could figure out ways, there are some structures. I showed you an example of one set of synapses. These are special cells in the hippocampus that are going into the granule cells, and those synapses in old animals become stronger. And so the question is, if you try to, that's presumably an adaptation, so we're working on agents that can cause synaptic strengthening. This is really simplistic. I'm not explaining it very well, but if we can facilitate, when you're learning something, if we could facilitate your synapses to become more strong, then it's possible that we could affect memory powerfully. And we do have animal models that show that. It's just getting these things into clinical trials that's difficult.
How does early retirement and mentally engaging hobbies affect memory loss?
Wave at me.
Oh, sorry. How does early retirement and mentally engaging hobbies affect memory loss?
Are you up there? I can't see you. Okay, oh, hi. Okay, how does early retirement and...
mentally engaging hobbies.
Oh. I want to get the word retirement out of the vernacular. You know, if you start a new chapter of your life, that should be renewal, right? You should be engaged in something else. I'm quite adamant about this. It would be, I think we need to get the concept of retirement, make it go away, because people that maintain an active lifestyle and maintain their social contacts and so forth, those are the people that do really, really well as they grow older. So I think active engagement and not wasting all the wisdom that's out there is probably the best approach. So if you retire early and then you start doing something else, your hobbies or whatever you want to develop, that's great. Yeah.
One of the figures you showed demonstrated correlation between brain health and physical activity.
Yes.
We would like to think that that means that if we're more physically active, we can improve brain health. How do we know that the correlation isn't the other way, that smart people are more physically active?
Well, I can answer that one way. It's an experimental design issue. That is, we start out with age, IQ, education matched people, and we put people into training programs, and we show that the ones that are being more active also show improvements in cognition. So that's the way that's been shown. Now, I'm not going to lie to you. The activity literature, the aging of physical activity literature is very difficult because while your your muscle and everything responds very well, your brain doesn't show like huge changes in or benefits in cognition, but it does show some small changes. So brain health is good. Keeping inflammation down, we know, is good because that means that you're not on the trajectory to Alzheimer's disease. But the changes are relatively small, but this is not an excuse not to exercise. You really do need to exercise.
Could you comment on white matter changes and the aging of the brain?
Yeah. So he's talking about those axons, those connections from one cell to another, and they have this wrapping around them that it's lipid, and that's the white matter that helps the electricity conduct much more quickly. And there is a good large body of evidence suggesting that there are white matter changes in aged brain. I don't think we know the reasons for sure, but I can tell you what I believe. I can't tell you that I know this for certain, but you know the synapse loss that I talked to you about onto those granule cells? Well, there's a change in the white matter going into the granule cells, but that's because of axon pruning. So the synapses are being lost and they're being pruned. So there is some loss. In Alzheimer's disease, you see widespread white matter changes. In Parkinson's disease, you see the same. So there are diseases that clearly show, MS, of course, show white matter changes. And it's so important to keep those connections as intact as possible. And the timing, so the fat around the axon, it makes, it confers conduction velocity. So timing of signals are also really important. Hi.
Good evening, Dr. Barnes. First off, I want to say I really enjoy your presentation.
Thank you.
And the question I have is about the selective cell loss. Now, you went over a number of ways of maintaining good brain health, but what is the criteria for the selective cell loss that does occur?
So we don't lose very many cells, in fact. And so there's a mathematical methods where you do brain cell counting. And this is after death in humans and animals and so forth, and you don't see widespread cell loss. Now, there is atrophy in the frontal cortex. So in the hippocampus, it's one story. You see almost no cell loss. You see loss of the connections between cells. In the frontal cortex, it's a bit different. So you actually are probably losing some cells. So why these different regions show, some do and some don't, I don't know. But the frontal cortex is interesting because it's a part of the brain that evolves last. So we and non-human primates have huge frontal lobes compared to rodents and other mammals. And it seems to be the part of the brain, well, it evolved last, but it seems to deteriorate more quickly. And actually, it develops the most slowly. So we're not completely myelinated. We were talking about myelin. We are not completely myelinated until our 20s in the frontal lobes, especially males. There's differences in males and females. Males aren't fully myelinated until 25 or so. Anyway, never mind. But that we seem to maybe this is a more vulnerable part of the brain. You see memory changes that reflect changes in those areas sooner. And I can't tell you why. It just, I can't tell you why, but it's interesting observation. Hi. Yeah.
Hi, thank you, Dr. Barnes. Wondering if you could say a word or two about the research on poverty and the aging brain.
One of the biggest challenges I think people that like me who think about aging have to face is that we start to determine our cognitive aging trajectory in early life. Until we start to deal with good nutrition, social environment, and so forth early on, we're really never going to be able to catch up as fully as we could if we started in the beginning. We need to, start that trajectory out in the most positive way possible. It's much easier to do that in early life than to remediate some way part way through. So if there's, this has got to be social policy. This has got to be social will to implement this. And I admit, I focus on the older individuals, but really, it's best if we could start in the schools early, say, even things like mindfulness training and so forth, to reduce stress early on. Exercise. I have an exercise study that I want to tell you about. That there's a huge experiment. About 100,000 people were examined on their exercise and their memory of their exercise. And then were tested cognitively on electronic tests and so forth. And what they found was that the highest correlations between when you did your exercise and how good your cognition was later was in the teenage years. So that you're extraordinarily active during teenage years, your cognition tended to be really good. So really active at any age was you're better than being sedentary your whole life. But even things like exercise can have a huge impact earlier on rather than starting later. But it's, yeah.
Thank you.
Hi.
Hi. Oh, sorry. Yeah, I have two questions. One is kind of straightforward. The first one is Tai Chi somehow really special, or yoga, anything else like that would be similar? And my second question is, are stem cell therapies effective in compensating for deteriorating brains?
So in terms of yoga, and there's just never been a study that has looked at the immune system and yoga, and so I can't answer that directly. My feeling is that any stress reduction method, and maybe yoga is going to be yoga for you, or quiet meditation. For me, it's active meditation. My walks up and around my waterfall behind my house in the morning to watch the sunrise. You know, everybody has a stress reduction method that probably works better. And so to prescribe something for any of you, I mean, it's going to be different what works for me than what works for you. So if it seems to calm you and focus you, use it. If yoga is good, you should use it. And the other one, see my memory, my working memory, I don't know, about stem cells. Yes, the problem with stem cells, as we know it, for Parkinson's disease, we know the exact nucleus in the brain that's responsible for deteriorating and giving the Parkinsonian-like symptoms. So some of the therapies that have used stem cells to replace these dopaminergic cells, this kind of special kind of cell in the brainstem, has been quite successful in changing, modulating the symptoms. But any kind of sticking stuff in the brain in various places and hoping it to work. You know, I think there will be therapies that come up that aren't as invasive as that. But stem cells are going to be an important part of medicine in the future, but it's still way out there from my perspective. But hopefully, I mean, we don't have a cure for Alzheimer's disease yet. So it's so tempting to think, well, should we be injecting stuff in the brain and so forth? But the word isn't in yet, I think. The word isn't in.
Thank you for your nice talk. I just want to be a little bit critical because it seems like everybody's not really looking at you from a critical point of view. I'm a non-medical researcher, so on the other hand, I'm still a layman from the science in the side of medical research. So I'm wondering why some things suddenly change conclusion-wise. For example, one day they're saying coffee is bad, another day coffee is good. And even in your own research, you said when you were doing in Carleton, the dendrites was certain kind of structure and it goes down as you age, but now it doesn't. So why did you have that previous result? Is it because you had bad experiments or bad instrumentation? Is it better now?
Well, I didn't contradict myself with the Carleton study, so I'm not sure what.
No,
what
you
mean
had a
by
dendrite circle, dendrite structure that was supposed to get worse as you
I went
age.
to the library, and that was the zeitgeist then. That was not my work. This was a
somebody
study
else.
from somebody else.
Yes.
And this is what's thought. And that picture is still in some textbooks today. It makes me furious. Anyway. There's no change.
Okay.
It's just wrong. But the caffeine, yes, there have been various reports, but if you do a meta-analysis of all the studies out there and clinical trials on caffeine, the net result is that there's an association with stronger cognition. And that association, we think, has to do with its anti-inflammatory properties, partially at least, for caffeine. It's still being studied, but if your stomach can take it, I think, it's part of the dietary supplementation, olive oil, omega-3 fatty acids in fish, things like this. Red wine, remember that?
Not too much of it.
And actually, I might just mention this for non-wine drinkers. Ethyl alcohol is a free radical scavenger. These free radicals are toxic to the brain. So even some alcohol in the form of ethyl alcohol, you don't get all the flavonoids, but you're getting this antioxidant activity as well. And it's been shown that those people who do not drink any alcohol whatsoever tend not to live as long as those who do drink some alcohol. But did I just say that? I think I just said that. Uh-oh. I'm fine. It depends on the audience. I don't care. It depends on you.
If you want to just let them know, we'll take 2 more. We've got
So-
2 people in line.
Okay, so 2 more, and then I'm thrown off stage here. So, okay.
Right up here. In order to improve the synaptic strength or connection
Yes.
and the plasticity, the learning that we need to do, the activities we need to do, is there a difference from what you understand in your research between an activity such as I do the New York Times crossword every day, but it's a habitual thing, as opposed to engaging in new learning?
Yeah. So let me answer a couple ways. The first is that if you really enjoy your crossword puzzles, you should continue to do it, and if it challenges you. But you shouldn't do it as a prescription to do it. And if the brain training games and so forth, my colleagues in neuroscience developed many of these, but the word in general in the scientific community is that if these brain games are taking you away from playing with your grandkid, taking a walk with your friends and being social and so forth, then you should opt out of the brain games. It'll save you money, and enjoy being social, because both exercise and socialization have a large impact on cognitive health. I probably didn't answer all your question. Learning something new. Oh, yes. And however, the retirement question fits into here nicely too. So taking on something new. Say you have left the piano for 40 years and you go back to do the piano, or say that you get to follow your passion in art and you start watercolors again, or you learn a new language. That is really good. But I don't think it has to be necessarily new. You just have to have a purpose and have to enjoy it. It's something that you can stick with. And really, crossword puzzles are fine if you really enjoy it, but you don't need to do that for good mental health. And brain training, what most of what we know about that is that you get really, really good at what you practice on a lot, but it doesn't generalize to other memories. And so the golden bullet for the perfect cognitive training system would be if it generalized across all memories. So you could do this, and then all kinds of memories changed. But that's not what we find. It's very specific. Okay.
Hello, Dr. Burns. I was wondering if any of your studies encompassed brain structure function in people from different cultures or different regions in the world, and how their hippocampus region would be smaller or larger depending on the area that they live in.
The one thing I can say is that that we do know, and know that hasn't been directly studied, but my guess, if you have peoples who navigate a lot, not by their GPS, but by having to know their way, like orienteers or something like that, those people's hippocampi would be bigger. In fact, Eleanor McGuire's, those showed that these taxi drivers have bigger hippocampi than people that don't drive taxis. Now, this was before GPS. She did this when they had to study for 3 years the streets of London, the one-ways and all that stuff. It was a big deal. So yes, the size of the hippocampus does change depending on whether you exercise it, essentially.
It's my great pleasure to be thanking our guest speaker, and I do so with the privilege of representing the Faculty Association, but I also, I am sure, represent the president and the university who are partial partners with us in this exercise. We heard earlier how this originated, and this speech tonight has demonstrated, as I think one other person mentioned, in fact, you mentioned, the sheer power of this lecture series. From time to time, I know for the people organizing it, it is a huge endeavor. It's a high-risk endeavor. Is it going to work? Are the speakers going to be as outstanding as tonight's speaker? But sitting here and watching and listening, we know it is just a wonderful experience and something that we all are very privileged to be part of. We are, all of us, and particularly the Faculty Association, very, very grateful to Professor Barnes for coming here. She not only speaks to us tonight, she met with researchers on campus today. Tomorrow morning she'll be meeting with students, and this evening, after tonight, the speech, you will be relieved to hear that a group of us will be taking her back to ensure that she has at least one element of the essential requirements for all of us as we age. Would you please join me in thanking her very much for the There are a few topics that could be more engaging than tonight's topic, without a doubt. I alluded to the work that our committee does, and I'd like to thank them all individually, but I also would like to thank their chair, Jasmine Habib, who has been with the committee for at least 6 years, I believe, and has chaired it for at least 2 or 3. Some of us could not remember. It's a long time. And this is, as I said before, an extraordinary endeavor that she took on. I think she could blame her dean, it was the previous dean, I believe, for saying that it didn't involve much. But those of us who work with Jasmine periodically and see the build-up to this presentation know how much work and devotion she has committed to this. And on behalf of all of us, I would like to thank Jasmine, and we have a tiny, tiny token to thank her for everything that she does. Thank you. By the way, she's stopping doing this. That's why we're thanking her so much. I thank all of you for coming out tonight. This is a wonderful event. It's nice to see you back in Hagey Hall, and we hope to see you again at the next lecture. And everyone, enjoy your evening, the rest of it, as well as we will. Thank you.